Have you ever wondered why cells divide? You know, there's a lot of good reasons for it. And different cells divide differently. However, we find that when we study the cell cycle that there are literally consistencies throughout practically every kind of cell. Now, once again, we have to make a special niche for our prokaryotic friends. With their one chromosome and their singular chromosome and their lack of compartmentalization, they're going to do cell cycle and cell division a little bit differently than you and I. I want to talk to you about the eukaryotic cell cycle. I want you guys to get a good feel for why this happens. Now, one of the nightmares, it's so often that when I talk to my students and I ask them for a little retrospect, many students will say, man, in high school, I never got cell division. And then you ask them in college, and in college, I never really got cell division. And then you talk to teachers, and they say, I had to teach cell division 148 times before I really got it. And I don't get it, why you don't get it, why it's so hard. No, actually, I do get it. Because I remember back when it was a struggle for me. And something I've learned over the years is understand the process, and you'll get the words. Okay? Just a little advice from an old guy here. If you get the process, it's all going to make sense. And you're going to see, as we progress through cell divisions and two particularly different types of cell divisions, that your basis is learning the eukaryotic cell cycle and what happens, and then it all stems from there. Well, let's start out with what's important. If you were going to divide a cell, what's important? Well, the first thing you would probably say is, why bother? Why bother having a cell cycle? Well, I think you know a little bit about this story. Why don't cells get to be giant? Well, cells don't get to be giant because they depend on their cell membrane for input of nutrients and for output of wastes. And it's a simple question of geometry. Volume increases proportionately faster than surface area. So the cell will get proportionately more volumetric, whereas its membrane, so that's going to, and the mathematical thing is it grows on a cubic basis, whereas the membrane only is a surface area. So if you just plug in some numbers, you find out that, wow, cell volumes get bigger a lot faster than cell surfaces. It becomes a volume to surface area ratio. Doesn't work. So cells can't get giant, which is a good thing for people who swim in lakes because amoebas stay on the bottom and they're tiny. Okay, but let's move on because, all right, so now we know why it has to happen. What would you want to do? Well, if you're going to make a cell duplicate, you have to make sure that cell can function. Think about it. Say I want to make a new skin cell, okay? There's a skin cell, and there's its nucleus. Now, if you want that skin cell, or this precursor to a skin cell, to be and to give rise to new skin cells, you have to make sure of something. You have to make sure that the two offspring, if you will, of that cell are going to be able to do a job. And what's its job? To be a skin cell, not to be a liver cell, not to be a sperm or egg, to be a skin cell. So therefore, we have to make sure that we get the genetic information that was in this first one, and we make sure that it goes into this one and this one. So we have to make sure that our genetic information gets in there. And so what has evolved over the course of the millennia, more than millennia, over the millions and billions of years that life has been around, what has evolved is this idea of before we can replicate a cell, we have to get its genetic material organized. So organization, if you think of compartmentalization of cells, it's an attempted organization and a good attempt. Well, there's even organization within that organization. A cell must organize its genetic information before it can pass it on. And here's the thing. Not only must it organize it, but it has to double it. We know that. This is what we're going to lay out for you over the course of this discussion on the eukaryotic cell cycle. How does the cell organize its genetic material, and how does it pass it on after doubling it? Well, let's take a quick look. Let's take a look at what happens to our genetic material. Now, I keep using the word genetic material because I want you to think. What is the genetic material? DNA. And what we know about DNA is that our cells, to organize DNA, go through a series of steps called packing. And we go from what is normally found in the nucleus of a cell called chromatin. And chromatin is DNA wrapped around proteins called histones and non-histonal proteins. And what happens is we go through a series of steps where this coils and coils and coils and coils and goes through a series of continued coils until eventually we get what we like to refer to as a supercoiled strand of DNA, which eventually ends up in this thing here called a chromosome. Now, we're going to talk a lot about chromosomes as we move through the cell cycle, but there's a couple of things I want you to understand. A chromosome is literally 2 molecules of DNA. You see, chromosomes only exist when the cell cycle is occurring or when cell replication is occurring. So what we just did was we went from a state called chromatin, which was that loosely packed DNA, to a chromosome. But even more importantly, we did something else. If you know anything about molecular genetics, you know that DNA can replicate. So in between this idea of chromatin and chromosome, we have doubled our DNA. And there is literally 1 molecule of DNA on the left side of the chromosome and 1 molecule of DNA on the right that are identical to each other. So we've accomplished our goal of doubling our DNA and organizing it. And what did I say is important? You want to make sure you organize that genetic material, and number 2, you want to make sure you get it out to the new cells that you're making. Which brings us to an overview of the cell cycle. And let's talk about what that means. The cell cycle is what a typical eukaryotic cell goes through. We've divided it into phases, excuse me, so that we can have a picture and have something to talk about. And the bottom line is, I know everybody thinks, oh my gosh, we're going to learn about this hard thing called mitosis. But if you take a look at the life of a cell before dividing, we're going to see that about 90% of a cell's life is in this green section here called interphase. Okay? And indeed, interphase is divided into 3 phases itself. Okay? We have a phase called G1, which stands for gap. You know, we've talked a lot in this course about cell biology. We've talked a lot about how cells function. We're going to talk a lot more about how cells function. Guess what? Right there, G1. We have been G1 experts. But then, at the end of G1, the DNA is duplicated. Okay? And that's when the cell is getting ready to do what I showed you a second ago, split into 2. And now we get a DNA duplication, a replication of DNA using enzymes like DNA polymerase. Did you ever hear of that? Maybe. Okay? And so our DNA is going to replicate. And now the cell is ready to divide, but it's got to grow a little bit more. So it goes, after S, DNA synthesis, it goes into gap 2. So when does it divide? Right here. Only 10% of a cell's life is spent dividing. Obviously, the field of biology spends an awful lot of time investigating what happens in the phases when a cell is doing cell things. But it's very important for us to now spiral in and take a look what happens in this small little section here, which we're going to call cell replication.